All right, so in 12.3, we are looking at how the cell cycle is regulated, and there's a variety of mechanisms in place to help control the cell cycle. But fundamentally, it comes down to a molecular control system, a series of molecules inside and outside of the cell that are going to help control the cell and its division process. And it really varies. How often a cell divides really varies based upon the type of cell we're talking about. So when we talk about the cell cycle, all the things that occur within the cell cycle, the sequence of events, are all controlled by something called the cell cycle control system, and it is pretty complex. We're going to, kind of talk about the basics to try to understand, at least a couple examples of how it's controlled. But that cell cycle control system is regulated by internal and external controls. So internal molecular, external molecular control systems. Now, when we talk about the cell cycle, we also have to mention that there are checkpoints, in the cell cycle, just like there are like traffic checkpoints on holiday weekends and things like that to catch people who shouldn't be driving. Well, there's also checkpoints in the cell cycle to catch cells that shouldn't be dividing. So the cell will stop at these checkpoints until it receives a go-ahead signal. And so if we look at this image, it is showing you the three major checkpoints of the cell cycle, the G1, the G2, and the M checkpoint. The G1 checkpoint, we believe it to be probably the most important checkpoint of the cell cycle. If cells receive a go-ahead at G1, we believe that they typically go ahead and finish the whole process, make it through G2 and the M phase and divide. Other cells that don't get the go-ahead signal at G1, they actually exit the cycle and enter what we call the G0 phase, which is a non-dividing state. These would be cells like nerve cells that don't divide once they've been developed. Our liver cells are usually in a G0 phase. There are some cells that can be called back from G0, and the liver is an example of that. When the liver undergoes serious damage, it can be enticed to divide, but typically once a cell is in the G0 state, that's where it stays. And so here again is another diagram, the G1 checkpoint. If you get a go-ahead signal, that cell will go ahead and continue the cell cycle and most likely end up dividing. If it does not, it exits the cycle and sits in a non-dividing state called G0. So the question then is, all right, what are these go-ahead signals? What is the cell receiving to tell it it's time to divide? There are really two kinds of regulatory proteins that are involved in this process. We call them cyclins and cyclin-dependent kinases, otherwise known as CDKs. Now, these things rise and fall. Their levels rise and fall with the cell cycle. That's why we call them cyclins. And there are lots of different types. But the one that we use as an example here in this text is called the maturation promoting factor, or the MPF cyclin. It is actually a cyclin CDK complex. So it's a cyclin and a cyclin-dependent complex together. When they're together, we call it MPF. Okay? When those two things come together, that is the point in the cell cycle where the cell is ready to move past the G2 checkpoint and enter the M phase, right? The maturation promoting factor. Some people remember it as the mitosis promoting factor, because when that cyclin CDK complex is together, which is a cyclin and the cyclin-dependent kinase, when they come together, these very specific two molecules forming that MPF, the cell will move past the G2 and start mitosis. And you can see here in this diagram how MPF activity fluctuates along with cyclin concentration, which makes sense if it's a cyclin CDK complex. So if you need cyclin to activate this CDK, then as the cyclin levels rise, it can start forming the MPF, that maturation promoting factor, and it will create that.
Excuse me, teachers, the IPI meeting is starting at this time, and this is Foster's room. Thank you.
Sorry. So the MPF will then be created and cause mitosis to happen. And then as soon as that cell starts dividing, cyclin concentrations drop very rapidly, as does the MPF activity. And you don't see MPF activity even showing up again until the cyclin levels start to go up in the next round. Here's another diagram of that same process, right? You can see that CDK is present in G1, S, and into G2. CDK is always present. It is a cyclin-dependent kinase. Kinase is a protein, right? So it's always in the cell. It, however, doesn't do anything until cyclin, which is being built up, in the cell. As we get through the S phase and into G2, we start to see the cyclin levels rise. And when the cyclin and CDK levels are at a certain point, they will interact and form the MPF, or maturation promoting factor. When that MPF is formed, that's the go-ahead signal for the G2 checkpoint. The cell will then enter mitosis and start dividing. And as it starts to divide, cyclin starts to break down. It's a cycle, right? It's a cyclin, which is a protein. And so it starts to break down at the end of mitosis. As cyclin gets degraded, the CDK no longer is activated, and the cell obviously is no longer dividing. So you can see how MPF levels are impacted by the cyclin buildup through the S phase and the G2 phase. So we also have, besides these molecular complexes like CDKs and cyclins, we also have internal and external signals at these checkpoints. So an internal signal would be something like a physical thing that should take place at that point. For example, kinetochores not having spindle fibers attached to them. If there are kinetochores that are not engaged by a spindle, they will send out a molecular signal that says, hey, stop, I'm not ready to divide. Because if it's not attached to a spindle fiber, then obviously the chromosome's not going to divide. So it can send out internal signals like that. External signals would be things like growth factors, like the platelet-derived growth factor, which causes human fibroblast cells to divide. Fibroblast cells are immature cells that can give rise to other things, and so this picture shows you without it, the cells don't divide. If you have PDGF in the agar, then the cells divide very well. Another example of an external signal would be something called density-dependent inhibition. I believe we talked about this when you guys were freshmen, but we call it contact inhibition. But basically that means if a cell is around other cells, if a cell is surrounded on all sides by other cells, if it's crowded by cells, it stops dividing. That's what normal cells do. But that's an external signal, right? It's getting a signal from the cells around it to not to divide. Also, animal cells will exhibit anchorage dependence, which means that they have to be attached to something in order to divide. If it comes unattached, or if it breaks free, then a normal cell will stop dividing because it's no longer attached. Right, so here's our example of anchorage dependence, right? The cells have to be attached to a substratum to divide, and as they divide, they will eventually surround each other on all sides. That's density-dependent inhibition. And if you scoop some of those cells out, the cells that surround the wound will divide until they are touching each other again. Now, cancer cells, on the other hand, do not show either of those two traits. They are not density-dependent, nor do they have anchorage dependence. So they can grow whether or not they are surrounded by cells, and they will also continue to grow even if they are not attached to a substratum.